This is your cell and molecular biology course. I'm your course instructor, Iniyatendra. The endoplasmic reticulum, or you can call it as ER, is a crucial organelle found in eukaryotic cells. It comes in two types, the smooth endoplasmic reticulum and the rough endoplasmic reticulum. Both are involved in important cellular functions, but they differ significantly in structure and the roles they play. In this video, we will explore these differences and understanding how each contributes to the overall functioning of the cell. In the illustration shown, you can see the rough endoplasmic reticulum in the left, where you can see small dots on the surface. They represent the ribosomes. And in the right, you can see the smooth endoplasmic reticulum, which lacks the ribosomes. The endoplasmic reticulum is one of the largest organelles in a eukaryotic cell. It plays a key role in the synthesis of proteins and lipids, detoxification, and transport within the cell. The endoplasmic reticulum is a network of membrane-enclosed sacs or tubes that extends throughout the cytoplasm of eukaryotic cells. Now let's see the structural differences between the rough and smooth ER. One of the primary differences between the smooth and rough ER is the presence of ribosomes. The rough endoplasmic reticulum is covered with ribosomes, which are responsible for its bumpy appearance, while the smooth endoplasmic reticulum lacks these ribosomes, giving it a smooth structure. In the illustration, you can see the cisterns and tubules of endoplasmic reticulum. You can see the surface of the rough endoplasmic reticulum covered with black dots, which represents the ribosomes. Down to that, you can see the tubules, which lack the ribosomes. These are the smooth endoplasmic reticulum. Now let's see the functions of the rough ER. The rough endoplasmic reticulum is primarily responsible for the synthesis and folding of proteins. These proteins are often destined for secretion or for use in the cell membrane. Rough endoplasmic reticulum is typically abundant in cells like pancreatic cells, where proteins production is high. So basically, rough endoplasmic reticulum is involved in the synthesis of proteins. Now let's see the functions of the smooth endoplasmic reticulum. Unlike the rough ER, the smooth endoplasmic reticulum is involved in lipid metabolism, the synthesis of steroid hormones, and the detoxification of chemicals. All these are carried out by the smooth endoplasmic reticulum. For example, in muscle cells, the smooth endoplasmic reticulum plays a vital role in storing and releasing calcium, which is essential for muscle contraction. So, generally, the smooth endoplasmic reticulum is related to lipid synthesis and detoxification of drugs and toxins. And also, in case of muscle cells, the calcium storage is carried out by these smooth ER. Now let's see the difference in case of location and abundance. The distribution of smooth and rough endoplasmic reticulum within a cell depends on the cell's function. As we have seen earlier, the protein synthesis, wherever the protein synthesis is high, you can see the rough ER uh, functioning, that is the abundance of rough ER. And wherever the lipid synthesis and detoxification is needed, the smooth ER is in abundance, such as liver cells. So for rough ER, you can keep the example as pancreatic cells, and for smooth ER, the liver cells where detoxification and lipid metabolism occurs. In the illustration, you can see the smooth and rough endoplasmic reticulum. Now let's see their role in protein and lipid production. One of the biggest differences between these two types of endoplasmic reticulum is their role in the production of macromolecules. Rough endoplasmic reticulum, as we have seen, it is involved in synthesizing proteins. In this illustration, you can see the rough endoplasmic reticulum involved in protein synthesis. The ribosomes is the site for protein synthesis, and these are transferred through transport vesicle to the Golgi apparatus, where there would be modification, and then they would be packaged, and then they will be released into the cytoplasm as secretory vesicle, and then these secretory vesicles will fuse with the cell membrane and release the proteins outside the cell. This is how the rough endoplasmic reticulum is involved in protein synthesis. The smooth endoplasmic reticulum handles the production of lipids and hormones. In the illustration, you can see the synthesis of lipid. It is given in a stepwise manner. First, in the cytosol, the fatty acids are activated by the attachment of a CoA molecule, which is the coenzyme A, which is a crucial coenzyme that acts as a carrier for activated SL groups, playing vital role in metabolism, particularly in fatty acid synthesis and oxidation, and also the citric acid cycle. And the second step involves the activated fatty acids bound to glycerol phosphate and are inserted into the cytosolic leaflet of the endoplasmic uh, reticulum membrane via acyltransferase, and then the phosphate is removed by the phosphatase enzyme. A choline already a choline already linked to phosphate is attached through choline phosphotransferase, and then the flipases transfer some of the phospholipids to the other leaflet. So these are the basic steps involved in lipid synthesis, and this shows how smooth endoplasmic reticulum is involved in lipid production. Now let's see the key examples of smooth and rough endoplasmic reticulum. For example, the rough endoplasmic reticulum in pancreatic cells is responsible for producing insulin, which is, as we know, a crucial hormone in glucose regulation. Meanwhile, the smooth endoplasmic reticulum in liver cells is vital for detoxifying harmful substances, thus helping the body to eliminate drugs and toxins. In the left, you can see the smooth endoplasmic reticulum of the liver cells. It is given that the liver cells are packaged with huge amounts of smooth endoplasmic reticulum because the liver is the primary site for detoxifying harmful molecules. In the left. In the right, you can see the release of insulin and glucagon from the pancreas, and the glucose metabolism is shown clearly. Now let's see the interactions between rough and smooth endoplasmic reticulum. Though the rough and smooth ER perform different functions, they often work together. For example, after proteins are synthesized in the rough ER, for example, after proteins are synthesized in the rough endoplasmic reticulum, they can be further modified by the smooth endoplasmic reticulum or transported to other parts of the cell. And both both rough and smooth endoplasmic reticulum are needed to maintain the cell integrity and uh, help the cell function and help in cellular processes. Now let's summarize the differences. So the basic difference is that the rough endoplasmic reticulum has ribosomes, while smooth endoplasmic reticulum does not have ribosomes. While the smooth endoplasmic reticulum does not, and uh, the rough ER produces proteins, while the smooth ER is basically involved in while the smooth ER is generally involved in the synthesis of lipids and it also detoxifies. By understanding how smooth and rough endoplasmic reticulum works, we can gain insights into the specialization of cells and their ability to carry out complex functions from protein synthesis to detoxification. Uh, these processes are vital for maintaining cellular health. Thank you for watching. See you in the next video.